Modern medicine relies on a massive, ongoing biological war between humans and bacteria. The opening offensive of this conflict was triggered by an oversight in a London laboratory. In the late 19th and early 20th centuries, humans were losing. A scratch from a rose thorn, a superficial battlefield wound, or a simple tooth extraction frequently resulted in lethal septicemia. Doctors understood germ theory, but lacked a reliable cure. Treatments were mostly diagnostic and palliative, relying on harsh antiseptics and highly toxic heavy metals. The primary goal for researchers was selective toxicity, the ability to administer a drug that would kill the invading pathogen without harming the human host. This inability to target bacteria while sparing the host left medicine in a state of desperation, searching for a biochemical mechanism that had yet to be observed. In September 1928, Dr. Alexander Fleming returned from a month-long vacation to his post at St. Mary's Hospital in London. Fleming had a brilliant mind, but famously disorderly laboratory habits. Before leaving, he stacked several Petri dishes of Staphylococcus aureus on his bench. Upon inspecting the discarded plates, he noticed an extraordinary anomaly. One of them had been contaminated by a rare fungus, Penicillium notatum. Looking closely at the culture, Fleming observed that the mold was secreting a substance that actively killed the surrounding Staphylococcus. It created a distinct clear zone where the bacteria had failed to grow entirely. Instead of simply washing the contaminated dish away, Fleming isolated mold juice. Recognizing the implications of this unhygienic anomaly gave us our first real weapon against bacterial infection. This side-by-side comparison illustrates the structural vulnerability that makes penicillin work. Human cells are bounded by a flexible lipid membrane, but bacteria require a rigid, mesh-like peptidoglycan cell wall to survive. The active molecule in penicillin features a highly reactive beta-lactam ring. This chemical structure acts as a structural mimic of the bacteria's natural cell wall building blocks. As a result, the bacterial enzyme mistakenly binds to the penicillin instead of its natural substrate. Once bound, the enzyme is permanently deactivated. The bacteria can no longer cross-link its peptidoglycan strands, bringing cell wall construction to a complete halt, while internal osmotic pressure builds uncontrollably. With the rigid exterior weakened, the internal osmotic pressure violently tears the bacteria apart as water rushes in. Because human cells completely lack these specific cell walls and enzymes, penicillin can circulate throughout the body in massive concentrations with remarkably low toxicity. Despite publishing his findings in 1929, Fleming's discovery languished in obscurity for 10 years because he struggled to isolate and stabilize the active compound. Turning mold juice into a drug required a massive collaborative effort. In 1939, a multidisciplinary team at Oxford University, including Howard Florey, Ernst Chain, Norman Heatley, and Dorothy Hodgkin, successfully purified, concentrated, and mapped the unstable molecule. The onset of the Second World War forced a massive industrial expansion. In 1942, the United States produced exactly enough penicillin to treat a single patient. 2 years later, by D-Day, production had scaled to 2.3 million doses. These historical metrics revealed the immediate military impact. In World War I, the U.S. Army death rate from bacterial pneumonia was 18. The initial laboratory accident provided the chemical blueprint, but it was the triumph of multidisciplinary engineering and global mass production that visibly shifted human mortality statistics. However, the massive success of this intervention immediately initiated an aggressive evolutionary response from the bacteria it was designed to kill. By saturating the human population and agriculture with antibiotics, we created severe selective pressure, fueling the modern crisis of antimicrobial resistance. Susceptible pathogens died, but any cell with a random genetic mutation conferring resistance survived and rapidly multiplied. Bacteria evolved precise countermeasures. Many produced beta-lactamases, enzymes that physically break open the antibiotic's chemical ring before it inhibits cell wall synthesis. Other pathogens, like MRSA, mutated their transpeptidases so the drug cannot bind, or deployed microscopic pumps to spit the medicine back out. These rapid biological workarounds reveal that the victory against infectious disease is not permanent. We are locked in a high-stakes, accelerating evolutionary arms race. The pipeline for discovering entirely novel classes of chemical antibiotics has largely stalled since the 1980s. To outpace resistant superbugs, researchers are being forced to look for entirely new paradigms. One approach involves bacteriophage therapy, which uses highly specific viruses to target pathogens without destroying the patient's healthy microbiome. Another avenue relies on CRISPR gene editing to selectively snip out the specific resistance genes within a bacterial population. Meanwhile, artificial intelligence is screening millions of chemical compounds in a fraction of the time it would take humans manually. This deep learning approach recently discovered halicin, a structurally novel molecule highly effective against resistant strains. The level of intense, cross-disciplinary innovation required today directly mirrors the mobilization of the Oxford team in the 1940s. Infectious disease control is a dynamic, unending struggle. Remaining ahead of these microscopic pathogens requires continuous global scientific collaboration, ensuring the medical advantage born from a messy dusk a century ago is not ultimately lost to evolution.